Good afternoon, everyone. So first is that it's very tough to speak after such an impressive talk. We will see. I will try to describe what we do at Vilmorin for pre-breeding, and you will see we are still really on the field, and not so much in the lab. Okay, pre-breeding program is a rather new program, so I will not show you so many results. I will just briefly present you the way we see pre-breeding. Okay, so the different part of my talks. So I will show you the importance of innovations in plant breedings. Then I will try to define how we see pre-breedings, and then I will describe what is I summarize as the 3 missions of the pre-breeder, which is enrichment of germplasm, trait discovery, and trait characterization. Okay, so the first example that I can give as a kind of innovative trait is that as an outcross crop, mainly OP varieties were used, and some public breeders find some CMS, and it helps towards the development of hybrids. The second example is, in fact, some private breeders in this case, they identify resistance to some diseases, in this case Alternariadosi, and they could develop varieties. So, in a way to say it, it's kind to show you how, because it was not named like that at that time, but it was pre-breeding, how it can change breedings and make new varieties. About the definition for pre-breeding, so one of the main definitions is to remove bottlenecks. So in fact, in crops, for all crops, it moves from what's just been shown. In fact, it moves from wild species to domestications and to breeding for modern varieties. And at each step, you see a huge bottleneck that eliminates a lot of diversity. So in fact, our modern varieties are just bred on, let's say, a subset of alleles. And one way to enrich it and to, let's say, to have a future genetic progress, it's to enrich in germplasm by using these, let's say, old varieties, even remote species, and it will be the scope of pre-breeding. Sorry. Another definition that we can give is to compare pre-breeding and breedings. I hope not too many breeders that will fight about this. First, we can say that the pre-breeding, that's what is red, and pre-breeding is in green, works on the more, we try to work on a larger basis compared to a light material in breedings. One of the easy aspects is that we focused on at least one trait, or few traits, but at least mainly one trait at a time, whereas the breeders has to focus on many traits to make a performing varieties. And for us, we look at a kind of highest value for the traits, whereas the breeders will try to make a kind of average value to find it at least commercial. So coming back to the missions, the first missions is the enrichment of our germplasm. As just been mentioned, the genetic resources is really the reservoir of variability for the future gain. So that's why it's very important. And what is it based for? It's based off cultivated germplasm, of exotic germplasm. I mean by that cultivated but not adapted to specific conditions, and also wild germplasm that is really rich in terms of diversity. That's for carrots, it's quite rich. I took as an example more or less the European type, so the Western carrot, and you see that all these kind of germplasm, so either Docus carota subspecies cultivated or not cultivated, wild species, are still to be used and integrated into the breedings, and other crossable Docus species can even be used. So now I'll try just briefly concerning the gene banks part to explain the way we work. So really the goal is to broaden the accessible genetic variability. One statement is that there is an increased complexity around seed exchanges worldwide. So in fact, what is our gene bank made of is made of our own accessions, of course, company accessions. We have good collaboration with public gene banks like USDI, Wary Crop Center, IPK, and also the Docus networks, with which we have a good and fruitful cooperations. We try to buy local exotic OPs, and we support collecting trips organized by gene banks, mainly for collecting wild Docus species at that time. Once we have some seed of it, the second step is really to propagate it, because we need two aspects in that. We need to characterize this material. So in fact, we need quite a bunch of seeds. And the second step is that we keep the seeds for long term, so we try for long-term storage. So multiplication is really the crucial point. And just to mention that we're also supporting the work done with the French Locust Network with Emmanuel. So once it's multiplied, you can describe it. So we do it a kind of brief description of the accession. Our goal is just to have a snapshot view. We describe different kind of traits, so really morphological trait, plant description, so foliage mainly, external root characteristics, internal root characteristic. We try to also using some flowering and some seed traits. It's more or less what is described in the hybrid guidebooks, but we focus on some other traits. For our accessions, we try to test them for key disease resistances. We will try to. And for cultivated accessions, we try to also evaluate them for key agronomic traits to be valuable directly for the breeders. The aspect of that is to know, if possible, the favorable traits that we want to use. But in the second step also, it's very important to know the unfavorable traits that we want to get rid of when an accession is interesting. One comparison with the public gene banks, in my opinion, maybe we can discuss about it, is that a public gene bank is trying to collect as much as diversity as possible. In our cases, we try to have a targeted and focused strategy. I can give you two instances. First is to take include the crop diversity. I took here, for instance, the Western Carrot program. That's from a public data, from Massimo, in fact. So that's mainly, let's say, the nonce program is based on the red circle there, and you see that you have a big bunch of variability that has not been exploited for this germplasm, so that's really the germplasm on which we will focus first. So that's our approach. And the second aspect is quite straightforward. It's also to take all info related to traits that is published or that is described. The second mission, after now we have this gene bank, is to look for trait discovery. That's one of basic jobs. So about traits, it's not, the whole title was new traits, but when we discuss, it's not really new traits. It's in fact traits that is already acknowledged by breeders as important to them. And we try to find new source of variability for these traits. That's mainly our goals. And we try to observe a wider genetic variation. And the work is done typically with breeders to set up priorities. Okay. One of the first steps for the trait discovery is really the development of phenotyping methodologies. So this work is done in collaboration with experts. I would mention two, typically the pathology teams, to help us to develop accurate and high throughput when possible. For disease tests, this is not so much high throughput. And now we're also more and more working for image analysis to get all kind of information around this. One other issue, I will show you some example, is that we're trying to have also to characterize like different biochemical compounds. So for the development of disease tests, also there we try to get accurate. We have some works, for instance, for cavity spots, where we try to have a kind of accurate evaluation for characterization of germplasm. Concerning the use of image analysis, so that's really to get accurate and high throughput. We're trying some pilot experiments for different projects, just like for characterizing root color. We also have some pilots for characterizing some flower characteristics, and also we try to use it for seed characteristics. As I mentioned earlier, we try also to have some biochemical characterizations, so either direct biochemistry using HPLC, for instance, in this case, or having indirect measurements using NIR techniques. And then, once you have this methodology for phenotyping, the second step is the evaluation of the variability across germplasm. I will try to describe two approaches. One is a kind of direct screen, and the second step is the development of integration population. It's in the case where you cannot assess the trait directly on the germplasm itself, but you need to come back to a normal carrot before assessing the phenotype. So in this case, that's part of the results I can show you that we started. In fact, it's just a screening, a wild population for disease-resistant traits. So that's what you see on the first graph. So you have a wide variation, and you see also within the population, you see the large bar, the arrow bar. The red bar indicates the susceptible control, and the green one, the resistance control. So what you do is that you pick out the best individuals, you just inbreed them and validate until the S2 stages. And what we have done then is that we have confirmed the resistance we have obtained. Watch out, in the last results, the pathology teams decided to reverse its scale. So now the higher it is, the more resistance it is. And what you can see, I tried to group by colors the lines, the S2 lines having the same origins, and you can find that you almost see some very high level of resistance. So it shows that screening wild populations, I mean, I will not convince Emmanuel about it, but it's very valuable for breedings. Okay. So that's when you can screen directly. When you cannot, typically when you want to evaluate some disease on the foliage, like for instance alternaria, you know that wild germplasm is annual, and bolting will prevent a good phenotyping system. So the goal is that we try to transfer by crossings to a kind of normal roots. So that's why we obtain after 2 cycles of integrations. And then we can assess the phenotype on a more, let's say, valuable phenotype. That's not yet a variety, but that's in between material where you can assess more accurately the phenotype. So that was the second mission concerning the trait discovery. And once it's discovered, then it goes to the characterizations. And the first steps for characterization is the integrations. Integration, it means by crossing into elite breeding lines. So the first goal that we can see is to evaluate the effect of the targeted trait in the elite genetic background. So you see the trait that you see, even if you improve, you're not completely sure that it's worked, because maybe the resistance is there because it's very small leaves or because it's waxy leaves. But in this case, once you come back to a normal phenotype, you can really assess it, let's say, accurately. And then it's, of course, a release of improved germplasm for breeders. The next step, we have not started that yet, but that's for next future. It's collaboration with the molecular guys that are developing nice tools for us. Is the genetic analysis of the traits of interest. So mainly our work is for the development of segregating population of panels, depending on which strategy you want to map your genes. Then you study some genetics about the heritability, about the QTL detection, and then try to develop markers for facilitated marker-assisted selection. So in the end, the pre-breeders deliver 3 things. So tools to support pre-breeding activities, improve germplasm, and genetic information. So just for the acknowledgement, I would like to thank the Limagrin Vegetable Seed Division that is financially supporting the jobs, and all the breeders that helped me to launch these programs, and all the technical team that really support daily the works. And one thing that I could add is that I would like to thank also all the public institutes I had discussion with for a fruitful discussion. Thank you, and if you have any question I can answer, I would be glad to answer.
Thank you, Arnaud. So let me start with a question in frame of your phenotyping program. Yeah. You mentioned you visit for CMS and flowering traits. Can you give more details for CMS? Would you prepare new sources? Or?
Not for CMS. In fact, what we try to look at, our most focus is to look at really more flower characteristics more than really CMS.
Okay.
So really more shapes or more morphology or color.
Okay, so we have time for questions.
Thanks for your presentation. From your perspective as an industry breeder, 2 questions about the public germplasm collections. First of all, are they generally useful? And second of all, are the wild relatives of carrot and I'm talking about Daucus carota wild, not other species. Are those from an industry perspective of particular interest? Do you work with those much?
To answer your question, I think the answer would be yes. I'm really convinced about the valuable. In fact, I don't have extensive knowledge of the characteristics because we just try to set up programs, so I have not really data to show. But I'm convinced that the genes of the future you can find in there, and just looking in a wider variability like the Daucus. In this case, the wild population, in fact, it was Daucus carota subspecies that we only focus on that. When you can see the valuable result that you can find, I think it's really open door for the future, and I think maybe I speak not in the name of the whole industry, but I think there is really good things to look at. Yeah. And I think it's just a matter of scale, because I think carrot breeders till now were really busy with all the tasks. Now it's really the matter to, we have a lot of molecular tools, we have access to a wide variability, so it is the step ahead to characterize and integrate this. The first question was, it was the exotic. Yeah. Yeah, that's good. No, I said yeah. No, but it's yes, no doubt about it.